So today's seminar, I will give you some brief introduction about my current research activity. So you know what do we do at Ohio State, and you can tell me what we can collaborate. All right, so that's the whole purpose. So, everyone sitting in the room must be familiar with the biomanufacturing mandate from President Biden. Now biomanufacturing become a buzzword. It used to be biorefinery or whatever, but now everyone call it biomanufacturing because it has so many impact using novel synthetic biology, bioprocess engineering, including AI, machine learning, and big data, and so forth. All the buzzwords, and of course, sustainability, environment, climate change. Okay, the CO2, circular bioeconomy, all those buzzwords involved here. And of course, I'm not sure this is a little bit over exaggerated. I said 1/3 of the global manufacturing output and will reach a market value of $30 trillion by 2030. But anyway, so everyone knows what is the bioprocess may look like. You start from the feedstock, different type feedstock, starch, sugar-based, or lignocellulosic waste, municipal solid waste, or food processing waste, or even industrial waste gas like CO2, carbon monoxide, syngas, and so forth. So different type of feedstock, they are renewable, sustainable, going through different type of treatment, or polysaccharide. You go through some enzymatic hydrolysis to break down into fermentable sugar, and then go through fermentation. Fermentation is the center part of the whole bioprocess. And you have different microorganisms, E. coli, Saccharomyces, those model organism, or non-model organism like Clostridium, which we work a lot in my lab. And you do strain engineering, synthetic biology to create robust cell chassis or factory. And one of the key today is TRY, titer, rate, and yield. You need to reach certain metrics in order to be economical for scale. So rule of thumb, you need to have productivity greater than 1 to 2 grams per liter per hour. Yield at least 0.5 grams per gram sugar, and titer ideally above 10% or 100 grams per liter. So those are the basic metrics you need to reach in your bioprocess. And of course, afterwards, you need to do some separation to get your product to the purity required for application. So the whole process need to be kind of in a complete scenario in order to make the product commercial. All right. So for that, you do the TEA, LCA, and those kind of stuff. And in my lab, in order to achieve the goal, biomanufacturing, bioprocess for chemical fuel production, we look at cell metabolic engineering. We do bioreactor engineering, develop novel bioreactor design, operation, and we do integrated bioprocess with in situ product separation to get the product more efficiently and at a lower cost. So biomanufacturing is a buzzword, but it's not a new thing to do today. Industrial fermentation, here I list a few historical or still today's typical bioproduct on the market: citric acid, ethanol, glutamate, lactic acid, lysine, penicillin, xanthan. Those are different type of product, primarily made by fermentation or microbial fermentation today. Okay, so the number for the capacity or production may be a little bit outdated, but give you some idea later in the large scale. And for different application, or you can imagine, is there anything you cannot produce by fermentation or by microbial process? Okay, so here I just list a number of different type of bioproduct you can make by microorganism, whether you use native micro, isolated from soil or somewhere, all genetically engineered. So basically, from food, drink, wine, beer, cheese, classical food fermentation to chemical like lactic acid, ethanol for fuel, all those things can be produced by microorganisms if you do it right. Okay, so even for the clothing, textile, paint, carpet, microbial cellulose, those can be made. So when we talk about engineering the microorganism, I only show you anaerobic pathway in E. coli. It can engineer depending on what end product. It shows a different organic acid, and the alcohol can be made by engineering E. coli or different microorganisms. This is in anaerobic fermentation, and in my lab in the recent years, we focus on butanol. We have also been working on propionic acid, butyric acid, lactic acid, and so forth. So in today's talk, I will focus mostly on butanol because it can be used as a drop-in biofuel and can be upgraded to aviation fuel or biodiesel. Another class of microbial product is the dicarboxylic acid, and there are many different dicarboxylic acid like adipic, those, and malic acid, succinic acid, and so forth. Those also can be made by microbial fermentation. People have done a lot of work to engineer different microorganisms to produce different dicarboxylic acid as a platform chemical or precursor to make polymer and so forth. Okay, so in today's talk, I will talk about the malic acid down in my lab. All right, first is about biofuel. GFA is Joint Bioenergy Institute, right? So biofuel. All right. So the reason we work on butanol is a 4-carbon alcohol has much higher energy content compared to ethanol. And it is more interesting, more challenging to make compared to ethanol. So for academic research, we want to do some challenging problem. Ethanol is a piece of cake, right? All right, so it can be upgraded to biodiesel and aviation fuel. Over more than a decade, in my lab, we have engineered butyric acid producing Clostridia tyrobutyrican to produce butanol, showing in here. So in this, the tyrobutyrican natively only produce butyric acid and acetic acid, not alcohol, but only need to clone one gene. ADHE2 is for bifunctional aldehyde alcohol dehydrogenase. You can convert acetyl-CoA and butyryl-CoA to ethanol and butanol, respectively. So it become a very promising and robust host to produce butanol. And a number of work have been done in my lab to engineer this with different pathways to use different substrate and so forth. And that was initially supported by National Science Foundation more than 10 years ago. And a couple of advantage compared to classical ABE fermentation using solventogenic Clostridia acetylbutyrican or Bacteriaceae that include this microorganism has a much smaller genome, easier to manipulate the pathway. And so it's easier to control the fermentation. And also, because natively produce high titer of butyric acid, it has very strong membrane. So it has high tolerance to butanol, can produce more than 20 grams per liter butanol. And ABE fermentation normally only about 10, 12 grams per liter. Okay. And these Clostridia, they don't have much bacterial fat contamination issue compared to solvent-genus Clostridia. Now, in historical industrial APE fermentation, one of the problem they face is the fat contamination. So we chose to work on this, and we continue to work on this. And there are a number of other researchers also joining us in developing cross-treated tyrobutyricum for butanol and the related product. More recent project funded by ARPA-E, EcoSyn Bio program. Because in the sugar fermentation, 1/3 of the carbon is released as carbon dioxide. So another challenge is, can we assimilate or reuse the carbon dioxide released in sugar fermentation and to further convert that into your final product? So to do that, CO2 gas fermentation, I know some folks here is doing gas fermentation, and you all know mass transfer is a challenge for gas fermentation. In this project, we convert CO2 through electrochemical reduction to fomate, and then we can feed the fomate easily together with sugar or lactate in the mesotrophic fermentation using the co-culture lactic acid bacteria and acetogen. Together, they can convert fomate and the lactate very efficiently, almost 100% yield to acetate. Then this is still the tyrobutyricum we've been working on. Tyrobutyricum can use both glucose and acetate to produce butanol. And later on, I will talk about a few challenges we are facing, that is natively produced butyrate, okay, and we need to convert butyrate also to butanol. All right. So stoichiometrically, theoretically, here are the reactions involved in this consortium fermentation. At the almost 100% carbon conversion, then the theoretical yield to get the butanol from 1 mole of glucose is very, very high. 100% higher than conventional APE fermentation or other engineered stream for butanol production. So if you say 80% conversion or efficiency, still has very high butanol yield, more than 0.5 gram per gram. Okay, so that's the current ongoing project, and of course the impact will be, you can get a much higher yield and zero CO2 emission, and you can reduce the product cost by more than 50% because you double the product yield from the sugar, you can reduce the product cost by more than 50%. So there is one challenge. The program called EcoSyn Bio. Now everyone knows to reduce the product, you need enough reducing equivalent to reduce the oxidized substrate and to reduce the product. Okay, so the overall reaction down here, we need additional NADH and the ferredoxin in order to complete the stoichiometry here I showed. Okay, so that is the challenge we are facing and we are working in this project. So to show you more in detail, that's the pathway in tyrobutyricum, the gene and the places involve NADH and the ferredoxin. Okay, so we have a few strategies trying to increase NADH availability for butanol production, because natively the organism produce butyric acid that require much less NADH, and the growth and the butyric biosynthesis has balanced ATP and the NADH. But when you force the organism to produce additional, more reduced product like butanol, you need to give them more NADH. All right, so here are a number of targets we have been working on. Say the ferredoxin NAD as the reductase that involved in the regeneration of NADH. Work on the HBD, the NADPH dependent beta hydroxybutyryl CoA dehydrogenase that basically replace NADH with NADPH. And the NADPH normally is used in biosynthesis for cell growth. Okay, so but there will be not sufficient NADH for the biosynthesis of butanol. But if we switch HBD to NADPH dependent HBD, we may make more NADH available for butanol biosynthesis. And we also work on inhibition of the hydrogen production. Some of you may be doing the gas to produce hydrogen. Okay, we wanted to inhibit hydrogen production, so we can use the electron to regenerate more NADH by using the missile biology or similar artificial electron donor or carrier. Additionally, I mentioned this tyrobutyrate can native produce butyric acid. All right, so we wanted to reduce butyric acid back to butyraldehyde and to butanol. So there are two ways to do it. One is to use AOR, the other to use GAR. Okay, aldehyde reduction acetyl reductase or NADPH dependent carboxylic acid reductase. Those are the two we are working on, trying to convert all the butyrate co-produced with butanol into butanol. One more thing is CAT1 is the transferase that is the key to assimilate acetate back to acetyl-CoA and for butyrate biosynthesis. We need to regulate this enzyme so we can balance acetate reassimilation or uptake by the tyrobutyrate and minimize the production of butyrate, because that's not the end product we want. Okay, so those are the gene target we're working on. And I show you a few results related to what I just show you. These are several different strain hosts with overexpressing ADHE2, different hosts, and this one with the CAT1 knockout on the genome. And you can see just overexpress ADHE2, you produce butanol, but still produce a lot of butyric acid, the open red circle. Okay. When we have CAT1 knockout on the genome, we produce very little butyric acid, much more butanol, but also produce a lot of acetic acid. Okay, so there is a compromise between acidic, butyric, and butanol production by manipulating the gene. Okay, and this one also has CAT1 knockout, but also we knock out SPA0A, and that produce also no butyric acid, but lots of acidic and butanol. Okay, then the table just show you the different mutant, the butanol yield. Okay, can be very different. Then we also create the mutant overexpressing the NADPH dependent HBD. Okay, and you can see all this mutant overexpressed HBD produce more butanol compared to the one without HBD, and that is because HBD use NADPH instead of NADH. Okay, so you can have more NADH available for butanol production, and you can see the yield now is higher than those. I mentioned we use missile biology to inhibit hydrogen production to make more NADH available for butanol production. You can see top row grows on glucose, bottom row grows on xylose with no MV addition with 200, 500, and 1000 micromolar MV addition. And you can see with MV addition, the butyric acid and or acetic acid production will reduce, and you will get more butanol production. You can see the next slide for the summary. So, for glucose and xylose with MV addition, you can see the yield of butanol increase, and for xylose, 0.3 is the benchmark, and we can go above 0.3 when we add MV. Okay, and one thing I like to note here is to use lignocellulosic hydrolysate contain both glucose and xylose. That's why we look at both glucose and xylose. Okay, and xylose is more reduced subject than glucose, so give you slightly higher butanol yield. but the xylose growth on xylose is slower than growth on glucose. So there's a balance between the growth and the production. Another thing is, in these cases, we see lactate is produced when you have MV addition at a higher level. Okay, that's because in the glycolysis, excessive NADH generated in glycolysis need to be consumed to recycle to NAD plus to support the glycolysis. And the fastest way for the organism is to convert, to use the NADH to reduce pyruvate to lactate. Okay, so normally they don't produce lactate, but under the situation we see some excessive lactate produced. Okay, so that is something we ask ourselves is, can we further increase butanol production by minimizing the acid, include lactic, acetic, and butyric acid? Because if we can eliminate all the acid, we should be able to reach our target of more than 0.5 gram per grain of the butanol. Okay, so that is something we ask ourselves. So this shows if the tyrobacteria can really reassimilate acetate, or if you externally add acetate as a co-substrate with glucose, can it be uptake and convert to the final product? Okay, so there are 4 panels here. Okay, with no acetate addition, this with delta GAT1 knockout, and acetate accumulation is significant because it cannot reassimilate acetate when you knock out GAT1. This is a different strain. Okay, you have butyric acid, but you can reassimilate the acetate because this strain has the GAT1 activity. Okay, so this is a complementary strain to the GAT1 knockout. Then you restore the acetate assimilation ability, but then you also restore the butyric acid production. Okay, so in the GAT1 is our target. We want to manipulate to use inducible or regulatory promoter to control the turn on, turn off of the GAT1 to balance the acetate and butyrate. Okay, that's ongoing. So those are what I just said. And I also mentioned we need to convert butyrate to butyraldehyde and butanol. So we look at the effect of overexpressing AOR on butanol butyric acid production. You can see here, one without AOR and the one with AOR. So with MV, you compare these two. With AOR, you see increased butanol production and slightly reduced butyric acid production. And here shows the pathway. AOR can reduce butyrate and the acetate to the corresponding aldehyde, then ADH will then further reduce to the alcohol. The cofactor requirement is shown in here. We try to use MV to regenerate the ferredoxin, reduce ferredoxin, but it looks like still very challenging. So we are working on alternative at this time. So that's what I just said. Another challenge for practical application is co-utilization of xylose with glucose in the fermentation. So if you look at the top here, this two, okay, the wild type, the xylose utilization was kind of inhibited in the presence of glucose. Although xylose by itself can be rapidly used by the tyrobacteria, but when the glucose is there, due to the CCR, carbon catabolite repression, it was repressed. But when we either overexpress the XY or TAB gene that is in the catabolic pathway leading to the xylose utilization, or knock out or knock down the transcription factor, the repressor XYR or HPRK, those will be improving the xylose utilization a lot. So you can almost have xylose utilization simultaneously with glucose when you either overexpress XYLTAB or you knock out HPR shown in the data here. Okay, so that's something eventually need to be built in the final engineer strength for co-utilization of glucose and xylose. All right, speaking about the process, currently we have project started with ABPDU as a collaborator. That is looking at the TRY and the stability for long-term operation. So in the process, we look at the fermentation and the separation integration to get the stable continuous butanol production. And conventional ABE fermentation using Clostridium acetobutyric, we run into a problem is the sporulation. After you run the fermentation for a while, this is done in a continuous immobile cell fermentation. You can get very high productivity, but after 2 weeks time, there are continuous decline in the production. You can see here, okay, the productivity and the production decline continues due to loss of the productivity or the viability of the cell due to sporulation. So how do we prevent or this kind of phenomenon is an issue we wanted to address. Even with in situ gas stripping for removing of the butanol, which is the inhibitor to the fermentation, shown in here, we also see decline in the production after a few cycles. Again, even if you continue to remove the butanol, still the sporulation still occur. Okay, so those are the issues we wanted to better understand and address in developing a high TRY butanol fermentation process with long-term stability of productivity. So this shows the repeated batch fermentation using the delta C81 ADG2 strain. The first 5-6 batch grow on glucose, second 5 batches using xylose, then using the mixture of glucose and xylose, then the next 5 batch using xylose, then we go back to the mixture of xylose glucose in the presence of NV at different or at increasing concentration. So overall, if you look at the repeated batch performance, that looks pretty stable. But if you look closely, after say 10, 11 batches, we see steady decline in the productivity. Okay, so this is using pyrobutyric and it's supposed to be much more stable than acetobutyric, the previous one I showed you. But still we see the decline in the productivity over time. So the question is why and how we can prevent this, right? So that comes to lifespan. Everyone like to live long, right? My mother just passed away 2 months ago. She was 99. We were hoping she can pass 100. Okay, so long life, long longevity is the desire for human being. And in the past, we don't think bacteria has lifespan, age, but today we know they don't have age. Aging is still a concern in industrial fermentation, even with bacteria. Okay, so this shows the motility, the survivorship or mortality can affect the decline of the cell viability or active or viable cell population. And that is especially a concern in say continuous perfusion or those are operation. Okay, and this shows when you grow the cell, different strain, and into the stationary phase, in the death phase, decline phase, different strain has different declining rate, the death rate. Okay, and the death rate is also associated with the lifespan or the survivorship of the cell. What contributed to this is something we have interest to understand. There are 2 types of lifespan. One is replicative lifespan. The other is chronological lifespan. Those will be affecting how active the cell can reproduce, for how long. Say India now has the most population in the world. They have high reproductive rate. So in the fermentation, if you have high reproductive rate, you can reach high cell density quickly. Okay, and if they have a longer replicative lifespan, they can reproduce more offspring for their lifespan. Okay, That's one type. The other type is chronological lifespan. This probably is more of our concern. In continuous fermentation, you want the cell to stay, not continue to replicate, but stay viable, productive for extended period. So that is require a good chronological lifespan. They stay viable, not going to age or lost the productivity. So those are the things we need to look at. What gene contributed to the different lifespan in the pathway? We know the lifespan is also dependent on the stress, the growth environment, and also on the genetic level. Okay, so that is the things we are currently working on. So in the previous slide, I show you the decline in the productivity. Okay, and how we evaluate the stability or longevity of the culture or the process. Now we develop a simple equation, the fraction of the production from subsequent batches. Okay, and for each batch, the probability or percentage of the loss of productivity due to aging, whatever. Okay, so that's the theta vector. So if you have 99.9% maintaining the productivity for each batch, and over the 20 batches, you still can maintain about close to 99% productivity. But if you have, say, 90% maintenance of the productivity after each batch, then quickly your productivity will be lower to only 12% of the original batch. So that can be a major concern in long-term industrial operation. So how do we improve the theta value? That means maintain the viability or vitality for each culture time. Okay, that's the challenge, and we need to understand what's the factor that into those. All right, so that's what I just said. So we designed an experiment for Clostridia, inoculate in serum bottle, incubate for 48 hours. Then we take 5%, 10% to inoculate to the next bottle. So in each transfer is one passage. We do it in 10 passage to see if the production change over different passage. And the change in the production is a refraction of the aging effect or the longevity effect. Okay, so we did one strand, delta HK DHG2, the other strand, delta CAT1, ADG2. And you can see with calcium carbonate as the neutralizing agent, this strand after the fifth passage continue to decline in butanol production. But this strand and the strand using sodium hydroxide can maintain their production over the subsequent batches. Okay, so this shows both the strain and the fermentation condition have significant impact on the productivity maintenance over a long period. So this just summarize those batch fermentation data. You can see for this decline and for this another strain, they can pretty much maintain the production of the butanol in subsequent passage. We only did 10 passage. If there's any notable change, we should be able to notice the difference. So summarize about the Clostridia fermentation, butanol fermentation. We engineered citalopenturican for different genes to improve butanol yield. And we were able to get high productivity through high cell density with cell immobilization and cell recycling. And we were able to get very high titer over 100 grams per liter through in situ gas stripping. And we also observed productivity loss over time due to the loss of viability or vitality due to stress and so forth. For the second part of the part is the polymalic acid, PMA, and the goal is to produce malic acid. And PMA, why? Because it's a polymer of malic acid, and malic acid widely used and can be a precursor to make a maleic anhydride as a platform chemical with very large market. And there have been many research for microbial malic acid fermentation, either using natively isolated strain or engineered strain, can get a relatively high titer yield productivity. However, none of them are in commercial application because the difficulty in purify the malic acid for chemical use, because other organic acid is co-produced and they are difficult to separate. All right, so that's the industrialization difficulty for microbial malic acid fermentation. So in my lab, since more than 10 years ago, we look at the black yeast, Basidium polulum. It's known to produce polulum, but it also produces polymalic acid as an actual cellular metabolite. It secretes the polymalic acid into the external. And the PMA is a polymer, can be easily separated from the fermentation process by adding alcohol to precipitate out. That way you can easily separate PMA from other organic acid. And then the PMA can be easily purified, then hydrolyzed to malic acid. So that allows the production of chemical grade pure malic acid instead of using direct malic acid fermentation. So we have worked on a process similar to what I have said earlier, but we can, because polymer can do ultrafiltration to concentrate, then do the alcohol precipitation, then hydrolysis to get the final malic acid at very high purity. And this project is currently still supported by USDA and NIFA. So we again work on the metabolic engineering, trying to further improve the PMA or malic acid yield from glucose. Actually, this project we work on a few things. One is to overexpress the gene in the reductive TCA cycle. It's here. That gives you higher malic acid yield compared to other pathway, the normal TCA pathway or glycosylate shunt. So the yield is much higher. Okay. And we also work on the knockout of the gene involved in the polulum synthesis. And we also look at the novel enzyme PMA synthetase that's responsible for the polymerization of malic acid and secretion. Okay. And also look at the xylose co-utilization pathway. But due to the time, I will only show you the engineering, the reproductive TCA cycle gene. So first, the black yeast is also non-model organism. Not many people have really engineered them. Okay, we are one of the pioneer group working in this area. So we have developed different transformation technology. Conventional for filamentous fungi or yeast, you may use the ATMP mediate transformation, but that is difficult to get the precise target to the genome. It's random insertion of the gene onto the genome. You have to screen, and screening is difficult, to find that the clone has the ability to overproduce the product. So we developed expression plasmid that can have very high expression level for the target gene. Okay, then we use this tool to study the overexpression of different genes in the reductive TCA pathway. Okay, and you can see when we overexpress the heterologous pyruvate carboxylase, that's from pyruvate to OAA, and the malic dehydrogenase from the OAA to malic acid. Okay, so we can see we can improve the malic acid or PNA production. Okay. On the other hand, we also overexpress like BAT, that's a transporter from the mitochondria for the malic into the cytosol. and that doesn't seem to improve. Okay, and we also did the double overexpression, say PYC and MDH. Okay, so we then look at one of the best strain co-expressing PYC and MDH in a repeated batch fermentation for high cell density. That's the fermentation kinetics. And we can get pretty stable PMA production from glucose. And if we compare the yield and productivity in the repeated batch, so this is only done for like 2 weeks. Okay, and we haven't had the chance to extend the repeated batch fermentation. So the same question, can we maintain the productivity or even increase productivity for a much longer time? That also go back to the question about the lifespan or longevity. And this just show we can easily get the final product PMA in the powder after alcohol precipitation. Okay, so that's a quick about PMA. So we produce PMA through the black yeast. We engineer a few target gene in the reductive TGA pathway to increase the yield. And we do the repeated batch fermentation with cell recycling that to reach high productivity. And with those improvements, we should be able to reduce the cost to around $1 per kilo. It's competitive for the current chemical on the market. And this just summarizes the comparison with other reported study. So this is what we get, tighter yield, rate of productivity. So we are kind of pretty good. Okay, in terms of TRY. Finally, I wanted to close my talk just to tell you, we have a newly funded NSF-funded future manufacturing project. It's future biomanufacturing. We specifically looking at the lifespan gene, how we regulate the lifespan to extend the productivity or longevity of the culture and maintain high productivity. And in this project, we also look at another bacteria, that's propionic bacteria. We have a collaboration with a company that are producing calcium propionate. Okay, and we are working on the cell recycling for extended, like I show here, extended production. Okay, so we're looking at the gene and how we can maintain the vitality for extended period. Okay, so that's pretty much my talk for today. And this is my current research group, 2 postdocs, 3 PhD students, and several research assistants, and my current research funding, and my collaborator. I didn't list Chang and so because we just started. Okay, and hopefully the list will be longer in a couple of years. Okay, so that's the end of my presentation. You have 5 minutes to ask me a question.
The lifespan was affected by sporulation. Was that something that was one of the lifespan associated genes, was the ability to turn on sporulation?
Yeah, the sporulation is a complicated life cycle event. And we have done a lot of study for sporulation in Clostridia and I didn't discuss those. Okay, and we found it is something we can deal with. We can minimize sporulation or delay sporulation process. But due to other issues like phage contamination and so forth, so we wanted to focus on Tyrobacterium. And Tyrobacterium by nature is less sporulated Clostridia compared to Acetobacterium. But we still need to deal with the stress response and the other longevity issue.
Have you studied whether there's any utilization of butanol in your engineered strains of the C. Tyrobacterium?
When you say use,
like any of the butanol that you produce, is it getting reused in the
you mean reused by the cell?
Yeah.
Occasionally we see the butanol peak and slightly reduced. Okay, mostly stay constant, but we didn't, let them sit for overextended period.
Sure.
Okay, so if we see some decline, most likely we think it's due to the analytical error. The other thing is when you sit there, maybe some butanol will absorb to the rubber or evaporated. We don't think the cell will utilize the butanol.
So I want to know, so how do you do the cell recycling? What method do you use for that?
Okay, there are two modes we use. Most people do the cell recycling with microfiltration or ultrafiltration membrane, but the membrane operation can be problematic for extended period due to fouling and other things. We prefer, just like in the current biopharmaceutical industry, they use continuous centrifuge. In the perfusion process, they use continuous centrifugation. That way, it's high throughput, and you can quickly concentrate and recycle your cell and let the medium perfuse and leave it. In the lab, we are not equipped with the continuous centrifuge for the small lab scale. The continuous centrifuge that with a good hygienic design is very expensive, and usually comes with kind of pilot scale or larger scale. Okay, so we have the cheap manpower. We use precious steel. We put we harvest the broth with the cell, put in the centrifuge bottle, put in the centrifuge, then do it carefully, and then get the cell and put it back. But we are in the process to make this process more industrial process, and the Jiedong's lab has the continuous centrifuge. We are going to start to transfer or translate that kind of process.
A final question is, so for the butanol you produce from the fermentation broth, how to separate the butanol from the broth?
Okay, so I briefly mentioned we use say gas stripping as a very effective way to get the butanol out. And we collect the condensate has the butanol concentration more than 10%, and the butanol solubility in water is about less than 8%. So they undergo the phase separation. So the upper solvent phase has the butanol concentration more than 80%, more than molar. So we can get very high concentrate butanol, even much higher than butanol. No ethanol fermentation. Ethanol, you talk about 10%, we can get 60 to 80% easily. So the energy for recovery, can be very, very low with those kind of process. So we work on both gas stripping and also pre-evaporation. Pre-evaporation is more selective than gas stripping. And I know some company I used to work with, they use pre-evaporation to get the butanol out from the fermentation process before they send to the distillation.
The first question is from Abby, and it's, how do you separate N-butanol from the fermentation broth? Does butanol have an effect on growth of other microbes in the consortia?
Yeah, okay, like I said, in the study, we have looked at different in situ separation methods, including adsorption, solvent cell extraction, and we found gas stripping is very convenient, easy to operate in the lab. And we also have looked at the pre-evaporation. And energy use-wise, pre-evaporation perhaps require the least energy for the butanol coming out from the fermentation broth. And the process has kind of semi-scale up to the industrial scale using pre-evaporation. And gas stripping, we believe, will be easy to implement as well. And we can combine gas stripping with pre-evaporation. But we haven't scaled up to the industrial scale yet.
There's another question from the chat from Justin. Was your yield that calculated for butanol from glucose assume no loss of mass due to cell growth? How would you limit this? Continuous fermentations are commonly used for adaptive laboratory evolution. So are you checking their DNA when your strains lose productivity to verify its sporulation and not just redirection of the carbon source due to genomic changes?
Different strain, okay. For the acetylbutyric, those gene for butanol production is native gene, so there's no change in the gene expression issue. It is more sporulation. We check those already. Okay, so we know sporulation reduce the productivity, and once the cell become spore or into the clostridial cell and the pre-spore stage, they lost the productivity. So that's the main reason we see the lost productivity. For the tyrobutyricin, they do not sporulate that much. Okay, so for the data I show you, the delta HK ADHE2, we believe under the calcium carbonate neutralizing agent, the lost productivity perhaps mainly due to the loss of the plasmid carrying the ADHE2 gene for that particular strain. Okay, but we also have data showing even with the genome engineered strain, they still lose, like I show you the later slide in the 15 repeated batch. Those are genome engineered delta CAT1 ADHE2, and we still see some decline in the productivity after 10, 15, after the 10, 11 batches. And the reason we don't know yet, but we are in the process to identify. Okay, it can be just a stress caused some genetic change or aging or whatever.
So this one comes from Young, and it's as you mentioned, if we want to increase the butanol yield, we need to decrease acetate accumulation. And then the question asker said, I think methyl viologen is using electron donor. And what do you think about the MV accumulation if you run the long-term operation?
Okay, the MV is more or less like the cofactor. Okay, so they are not consumed. Okay, so if we run the fat batch or continuous fermentation, continue to add the sugar, glucose, and continuously to remove butanol, then we don't need to add more and more methyl viologen throughout the process. We only add at the beginning, then they stay there. They will work for the entire period.
I think it's right on time. Thanks everyone to be here on online. Let's give a final thought to Dr. Yang.